My name is Lisa Steno-Biddle. I'm the CEO of Likarda and very happy to be here today. Thanks to AIM for ARM for organizing this meeting. This is our first ARM meeting, so I'm very happy to be here. In the past 2 days, we've seen some really amazing cell therapy technologies and breakthroughs discussed, and it's very inspiring. But we've also heard about some challenges, and one of those challenges is delivery of the cells, of sufficient numbers of cells, to the target destination to have the desired effect. And so how are companies getting around that? Well, they're starting with really huge doses of cells, or they're multi-dosing. What if you could take that cell therapy and mix it with a delivery system that would help you to target the cell to a localized area and slowly release cells in that area over a known period of time? And that's what we have at Likarda, is a delivery system for cell therapies. Our delivery system is customized, so we've learned that almost every cell therapy requires a different formulation for the encapsulant. Again, it can be delivered locally to the area where it's held in place, and as with other encapsulants, our work also protects the cells from the immune system. It's a novel platform technology, part of the manufacturing process. It's highly controlled, so we can control the size of the beads, the loading density of the cells, the duration of the bead and how it degrades. It's injectable, and as I said, it will hold the payload in the place where you're targeting. And most importantly, we start with biomaterials that are already known to be compatible. So things that the FDA has already cleared for wound healing, for cosmetics, as fillers or joint lubricants, we use those materials as our encapsulants. And that's really important because you've probably heard of other materials that will initiate a foreign body response as an encapsulant. And I give you an example here on the left. Those beads are destroyed by the surrounding tissue, and so are the cells that are contained within them. But our materials, because they're already, we use the term stealth, they're invisible to the system, and many of them are already native to the body. So you just don't see that foreign body response that you have to deal with. We have over 55 different hydrogel formulations that we've produced and tested, and we put them into buckets. So the first bucket is our short-term encapsulant. So here you need the cells to engraft, or at least to come in contact with the surrounding tissue. We have release mechanisms, so if you need the cells to be released in an area in a period of 3 days, we can do that. If it needs to be weeks, if it needs to be months, we can design the hydrogel so that it delivers the cells in that time frame. We also have durable hydrogels. Now we want to protect the cells from the immune system, and it's not the cells that are the active ingredients, it's what they're producing. So it's the exosomes or the cytokines, or if it's a genetically modified cell, it's whatever you put in the cell to create. And the cells are held and protected from the immune system. We also, because we know so much about the chemistry of these hydrogels, we also have long-lasting, fully cross-linked, cell-less microbeads for things like long-lasting cosmetic fillers and joint lubricants, and I won't be discussing those today. So first, just an example of our short-term hydrogel and one application. Our first application is osteoarthritis. I don't have to tell you how big this market is, and that the current care, the standard of care, is less than optimal. Most people with severe osteoarthritis will end up with a knee or joint replacement. You probably know someone who's had a stem cells injected into their knee. I have a friend, and it was a miracle for her. She didn't need a knee replacement because of it. But for other people, it has no effect at all, which is why the clinical trials to date have failed. Research in animal models tell us that that's likely not because of the cells themselves. It's because the cells don't stay in location long enough. When we weight bear on that joint, the cells are removed from the joint and cleared from the body rather quickly, even in less than 24 hours. This is a rat study of severe osteoarthritis, and we let the osteoarthritis go to a very long time point so that we knew that the animals were in fact osteoporotic. It's a destabilized medial meniscus model. And you see in the control case, we just gave them placebo. In the middle, at 6 weeks, we added stem cells. We injected stem cells into the knee. And it improved the outcome of the knee. The surface of the bone is less jagged. The red staining that you see is for new cartilage, so there's some cartilage there. The stem cells had an effect, but that's still a painful joint. Now, if we took the same volume of stem cells, or the same number of cells, and we first encapsulated them and then injected them into the knee, you see a completely healthy knee, a smooth joint surface, a lot of new cartilage, and that's a pain-free joint. And we can quantify that with the Orsi histology scale mechanism, which is what the graph is at the bottom. So now, an example of the long-term or the durable hydrogel. And in this case, we're targeting diabetes. So this is a dog model of diabetes. We are now in our large animal model. And on the left, you see the current standard of care for spontaneously diabetic dogs, so dogs that are family pets that have diabetes. It's twice a day insulin injection. Oftentimes, the dogs are getting human insulin. And you can see that even with a diligent family, this dog is spending a lot of time hyperglycemic. The green boxes are target range for blood glucose. We did a dose finding study, and that's what you see on the right. So this dog was getting 8 to 10 units of insulin every day prior to the first transplant. And again, the first transplant was a subtherapeutic because this is dose finding. And at that point, and this is just an IP injection or infusion with a small catheter, after the first transplant, you see that the blood glucose is normalized, and now the dog is getting 1 to 2 units of insulin every few days. We then got a second infusion and got the dog up to therapeutic dose. We stopped all insulin injections. It took about a week for the cells to figure out what to do, but at that point, the dog was now in normal range of blood glucose and required no more further insulin. Now, those cells in this study were obtained from cadaveric islets, so a donor source. We now are repeating the same study with a scalable cell source. So we have insulin-producing iPSCs. We call them epipses. So we have now encapsulated our epipses, and we have finished dosing one dog. And at 50 days out, she looks almost identical to the islet transplant. She is no longer on insulin. And today, this afternoon sometime, we should have dosed our second dog. So we're just getting started with this 6-month study. So a little bit about the company. I told you about the 2 indications that we chose just to highlight the short-acting and the long-acting encapsulant. But we have many other applications where we've partnered with other companies. They have the cell therapy, we have the delivery system. So from this table, I think you can see a lot of different applications that we are targeting, and the vast types and numbers of cells that we're using, anywhere from fully differentiated cells, adult cells, to multipotent stromal cells, and we can do all of that work with our encapsulants. But again, it's very important for me to say that if you hear of someone talking about they have an encapsulant and it works for all cells, that's not biology. Our experience is that every cell type needs a unique formulation. They need a unique microenvironment, and we can do that with our large library of hydrogels. So if we look at the competitive landscape, that kind of falls into 2 categories. Macro devices or macro encapsulants like ours, and then micro encapsulants like ours, and macro larger devices. So with microencapsulants, again, we're the only company that has a slowly controllable release formulation that releases your cells in a timed fashion. We're the only company that can customize the hydrogel to your cells and your application. And of course, it's an injectable payload. The microspheres are not retrievable on their own, but we are partnering with companies with devices to put our beads into their devices so that we will have retrievable options. We have a very experienced team running the company with decades of experience in both formal or traditional pharma, as well as in cell therapies, manufacturing, and QC/QA. And we have a very robust patent portfolio. Now, in the red and green, we have patents issued around our stem cell technologies, and I didn't really talk about that today. But the blue highlights what I did talk about, our hydrogels. Our CSS, the platform technology, manufacturing technology, has been, patents have been issued over 20 times around the world. And we also have patents for specific formulations and specific applications like the slow-release formulation. The CSS patent is good through 2035 at a minimum, so we have a long runway. So why am I here talking to you today? Well, we're looking for both investment opportunities and or investment partners and also cell therapy companies to partner with. So first, a little bit about the investment. We are in a Series A fundraising round right now. We are looking for our lead investor, and so the terms are still negotiable. But what will we do with this money? Well, we will complete our large animal studies. I talked about this dog study. For the osteoarthritis, we are also working on a spontaneous large animal model. So later this year, our product will be going into lame horses. And so the money will pay for that. Also, we need to develop commercial scale instruments, our microencapsulant technology. And so we will be doing that and expanding our preclinical manufacturing site. We'll bring in more expertise in manufacturing and regulatory, and we will grow our pipeline of cell therapies, partnering with other companies by adding a dedicated marketing and sales force. Now, for our partnering companies, the way we work with our partnering companies is we start with a POC project, fee for service. So we'll bring your cells in and design for you an optimal encapsulant or hydrogel. Then we move on to development, preclinical studies, and we intend to license then that hydrogel once you're thrilled with the results. What do you get for that as a partner? Well, you get a unique microencapsulant formulation that's built just around your specifications. It can be durable or short-acting, and again, that time frame for the release of the cells, you tell us what you want, we can build that. Your cells, while they're in the microencapsulant, will be protected from the immune system. And another thing that I didn't talk about was that we find that our microencapsulants are also very helpful in cryopreservation of your cells, so you'll get a higher viability if you're cryopreserving the cells after microencapsulation. It also extends the shelf life, even at room temperature, if the cells are first microencapsulated in an optimal environment. So I'm happy to take any questions. We do have just a little over a minute, and thank you for attending.